Buenos dias, and Hola from New York City. I'll be presenting some new data today, in addition to data that have been around for a while, which are the work of Alex Procht, a postdoc in my lab. And there's one of the most precise ways to manipulate CNS arousal is to anesthetize people. It's probably more precise than the sleep-wake cycle, and certainly much more pleasant than coma, which are the other two ways of doing it. So I'm representing Alex here today. So the theme of this week, engineering principles in biological systems, what that means to me is that we require knowing the elements, the concepts, and their quantitative relations to each other. And for me, in this field, the ultimate goal of a neuroscientist should be to achieve the precision of physics and mathematics applied to the CNS. And I'll present some new data today about generalized CNS arousal, but the way we used to do that in our lab was to explain an extremely simple sex behavior. The only way of being precise was to keep your problem simple enough that you could actually solve it, which we did, as you will see. And in that interest, I'll show three short stories. I'll talk about the concept of generalized CNS arousal, the concept. I'll show you an assay, and I'll give you a very brief view of the mechanisms that we know so far. And in honor of this week's theme, I'd like to emphasize quantitative analyses and also get your reaction and Partha's reaction to these quantitative analyses. And then, after maybe 2/3 of the talk, I'll completely switch gears and go to the simplest behavior that a mammal can show, which happens to be a very simple sex behavior, which is lucky because it shows how these vertebrate animals reproduce. And finally, just in a couple of slides, I'll make what should be obvious to you, mechanistic linkages between generalized CNS arousal and sexual arousal. The last time you took methamphetamine, you probably understood that there's a relationship between them. So about this concept of generalized CNS arousal, which I consider to be a primitive function and the most powerful and essential capacity of a vertebrate nervous system, is to say that this generalized CNS arousal is fundamental to all cognitive function. You can be aroused without being alert, but you can't be alert without being aroused. You can be alert without paying attention, but you can't pay attention without being alert, and so on up the cognitive food chain until we get to the higher aspects of decision-making, which are probably controlled by the cerebral cortex. Likewise for emotional function, whether you're expressing feelings on a lifetime basis, an hourly basis, or a minute-by-minute basis, arousal is at the base of it all. And so if you were to think of emotional expression as a vector, like in physics, surely the angle of the vector would depend upon what the feeling is and toward whom are you expressing that feeling. But the length of the vector, the strength of it, is going to depend upon generalized CNS arousal. And it should be self-evident that among all of the things that you do in the central nervous system to arouse the brain, that one thing must be deeper than all the others, and that generalized CNS arousal is what I'm going to be dealing with for the next few minutes. So given that waking up the brain is important, when something goes wrong with arousal pathways, all kinds of hell breaks loose. The most obvious, of course, would be the stuporous, the vegetative state, and the comatose states, but also the dementia of aging, the dementia of Alzheimer's and the apathy of Alzheimer's, attention deficit hyperactivity disorder. Autism, I believe, has a feature of social anxiety, which is an arousal problem. The last time you were depressed, arousal pathways were involved. All of these public health things, vigilance things in the military, shift work, dangerous occupations like preparing the fish or preparing the meat is the most dangerous thing that you can do. These toxicology questions and fatigue states, they all involve generalized CNS arousal. And from my point of view as a professor, the importance of this list is that you can think of more ways to get money from different kinds of disease foundations. Most of the time I've been going through this list, I've been talking about problems elevating CNS arousal, but sometimes the problems are in a regulated decrease of CNS arousal. And so, for example, Alex Procht in my lab would be concerned with the regulated decrease of arousal in the anesthetic state and sleep disorders. In the States, at any given moment, 15 to 20% of the adults will have some kind of sleep disorder. So those are problems as well. So given that these pathways are important, the concept of arousal, when I entered this field, seemed to have kind of a vague and a slippery character. You couldn't quite get a hold of it. And so what I wanted was an operational definition which is at once precise and complete, and also leads to physical quantitative measures, precise quantitative measures. And so I came up with the most broad definition of operational definition of generalized arousal that I could, which is that a more aroused animal or human being will be more alert to sensory stimuli, emitting more voluntary motor activity, and be more reactive emotionally. And we have an assay in a genetically convenient animal, that is the mouse, and I think almost all of the slides that I'll show you will be about mouse brain and mouse behavior. And so on the sensory side, we have tactile stimuli, olfactory stimuli, and vestibular stimuli. Those would be three sensory modalities which use different kinds of neuroanatomy. On the motor side, and that part of the operational definition, we're measuring the animal's behavior with 20 millisecond resolution. Yesterday we heard 536 million years, or maybe it's 636 million years. But today we're dealing with milliseconds, 24 hours a day, 7 days a week. And on the emotional side of the operational definition, you could say, what can you really count on for emotion in a mouse? And emotion in a mouse probably means sex and fear. But we can't do sex in this part of my talk because we want each animal to be in a cage absolutely isolated from the environment, untouched by human hands, everything computer controlled, and so we have to be dealing with fear. And therefore, the floor of this assay is a grid, and we can give electrical shock, and so we're measuring either conditioned fear or unconditioned fear. So we can assay the sensory, the motor, and the quotes, emotional, close quotes, 24/7 with 20 milliseconds, well, the movement with 20 milliseconds resolution. Now, at this point, we've got a concept, an operational definition, and an assay. And the question is, can we really prove that generalized arousal exists, other than our preconception that it must exist? So there's three ways of doing it. First would be the quantitative behavioral evidence. And over a period of years, we collected a lot of data in assays that have to do with generalized arousal. And using a calculation called principal components analysis, we measured that about 1/3 of the variance is accounted for by generalized arousal. And the other 2/3 would be, if you think of generalized arousal as an equation that has many terms in it, the generalized, if you have overall arousal as an equation with many terms in it, the generalized arousal term would account for about 1/3 of the power of arousal. And then all of the other things that we don't know so much about would account for the other 2/3. We could talk about sex, we could talk about hunger, thirst, fear, and many other, maybe some things that we cannot even name. Okay, so we have calculation evidence that generalized arousal exists. Also, breeding for arousal. I think you'll agree with me, you cannot breed for something that doesn't exist. Therefore, if we can breed for generalized arousal, it must exist. And so what we did was, we've been actually now through generation 10, and we do have significant separation. This is difficult and expensive because this is not like Drosophila eye color, where in a generation or two you can breed out or breed in a certain eye color. This is obviously a multigenic trait. We have significant separation, but there's still some overlap between the distributions. So we have calculations, we have breeding, and also, even as you cannot breed for something that doesn't exist, you cannot have mechanisms for something that doesn't exist. And so what I'm going to show you very, very briefly, this will be a rapid New York City-based rate of tour of three kinds of mechanisms: neuroanatomical, neurophysiological, and functional genomics, as to how generalized arousal is produced. Neuroanatomically, we have ascending pathways, and you probably have heard of all of these. You have norepinephrine ascending from the brainstem into the cortex, especially serving sensory alertness. You have dopamine ascending from the brainstem toward the limbic system. Dopamine, the simplest way of talking about it, the most parsimonious way, is that dopamine accounts for movements, directed movements toward salient stimuli. We have serotonin, obviously involved with emotion. Antidepressants often deal with serotonin. We have histamine, which is an arousal transmitter par excellence. If you took an old-style antihistamine to solve your allergies, then you got sleepy. That's because it's a histamine receptor 1 blocker. And finally, we have acetylcholine, which not only comes from the brainstem, but also from the basal forebrain, to invest the cerebral cortex in very complicated mechanisms that serve to increase generalized arousal. Some of the medicines that attempt to stave off, to delay the symptoms of Alzheimer's disease, deal with chemicals which preserve acetylcholine. Yes, Partha?
Just one small comment. I'm very pleased to note that you called dopamine something associated with arousal rather than reward, in keeping with concepts in sexualization as well.
So Partha is referring to the overinterpretation by the extremely famous Wolfram Schultz, who moved from Germany to Oxford, I think. Is that right? Yeah. And he managed to sell to the editors of Science that dopamine is a reward transmitter. And of course, dopamine is a reward transmitter. That's why we take cocaine. But the most parsimonious way of talking about dopamine is that it rewards, it enforces movements towards salient stimuli. And one way of making a stimulus salient is if it's a conditioned stimulus for reward. It's just a moment of thought will convince you of that. Now,
two questions.
Yes, please.
Just for
Nick.
clarification, okay?
Yeah.
So earlier you were referring to this behavioral essay you have.
Yes.
You say you can explain about 30% of the behavioral variance in terms of arousal. Are you going to show us some of that data as well?
No, it was published starting.
That's just a claim.
The principal components analysis was published first in 2003 in PNAS in a paper by Gary et al. And then it's been published, subsequent analyses have been published 2 or 3 times since then.
Right, so but then, so what are the operational definitions you have been using in that case to establish that observation? So that's just, let's say, the variability in the behavior, more directed behavior, just to understand what we're really talking about.
No, it was all arousal-related behaviors. In other words, speed of behavior, acceleration, not learning behavior, nothing complicated.
No, but it's fear-driven, right? So I guess we're talking about freezing or something.
Freezing would be one example of it, yes.
Okay.
Yeah.
Right.
The fear was part of it.
Right.
Yeah.
So then the second issue for clarification.
Oh, by the way, this other paper, the other main paper is by a guy named Zachary Weil, W-E-I-L.
Mm-hmm.
Yeah.
Right. So in some sense, for serotonin, histamine, and acetylcholine, you sort of summarized it a little bit, going more, saying, well, okay, they affect sort of general arousal in some way.
Mm-hmm.
But how can we still differentiate among these systems, just to be able to follow what's going on?
So just to repeat the question, since you're in the front row,
Sorry.
the question would be, I'm trying to talk about generalized arousal, but the question properly and accurately says, yes, generalized arousal is great, but aren't there some special features that the different transmitters specialize in? And yes, on the sensory side, norepinephrine would be more prominent. On the motor side, dopamine would be more prominent. On the emotional side, serotonin would be more prominent. Histamine is essentially a wake-up guy, and not specially sensory or not specially motor. And acetylcholine really supports the operation of the thalamocortical system. If we're talking about the dorsolateral nucleus of the tegmentum, then we're talking about thalamocortical systems. There's a high road to arousal and a low road to arousal. The high road would be the dorsolateral nucleus of the tegmentum feeding the central thalamus, which in turn wakes up the cortex. If you have a vegetative state patient in front of you, as Nicholas Schiff showed in Nature 2007, and you stimulate the central thalamus, it's possible to take that vegetative state patient and bring him into a state of functional cognition. It's like a miracle. Whereas the low road to arousal, probably the more evolutionarily ancient one, but Nick could tell us more about that, is in the basal forebrain. And when people have tried, as I said before, to deal with Alzheimer's disease, they're thinking more about these giant cholinergic neurons in the basal forebrain. So your point is absolutely correct, that each of these transmitters has 2 faces. It's like Janus, the famous illusion. 2 faces. One would be the face toward generalized arousal, and that's what I want to deal with, because I want to deal with a theory of everything, if you'll permit me to say that. Whereas there's also the more specific forms of arousal. Fear is not the same as sex, although they sometimes occur at the same time. Yeah. You remember that.
Don, can I ask a question?
Yes, please. Yeah.
So the medial reticular formation and the reticular activating system theory of Magoon, what's the status of that these days?
It's alive and well. In the history of science, you'll often see that things are so old that they're new. And Marazzi and Magoon, pioneers, published 1949 was one of their famous papers. Magoon, a book called The Waking Brain, published in 1954. I recommend it. It's only that thick. Yes, what I have done is I've tried to take the empirical data of Marazzi and Magoon and put them both in a strict logical framework and to use modern techniques and to study. They had very limited techniques in 1949. And also, people got distracted for two reasons. The first is that actually there were papers by Morrison and Talbot in 1942, and they opened up this field that I'm telling you about in 2012, but they got discouraged because nobody paid attention to them. Nobody paid attention to them because there was a small event called World War II going on. And so by the end of World War II, they'd gotten discouraged and given up. Then Marazzi and Magoon came along and provided the data which encouraged me to write a book about 2004 called Brain Arousal, which was really taking Marazzi and Magoon and trying to cast a new light on it. Some of the nicest problems for modern techniques are actually the oldest problems. Yeah, Giovanni Marazzi and Pisa, Italy. Now, everybody pays attention to the ascending systems, but many people don't realize that there are also descending systems which are very important for waking up the brain. Have you heard about orexin hypocretin? Tonight at the party, if one of you has narcolepsy, which would be a mutation, either two different groups called this two different things. Yanagasawa called it orexin, whereas Delicia calls it hypocretin. Delicia is a much nicer guy, so I always called it hypocretin. Or if you have mutations in either of the receptors for this, tonight, if somebody tells you a big joke and you're laughing like crazy, standing up, if you have these mutations that I'm talking about, in a couple of seconds you'll be lying unconscious on the ground. That's what narcolepsy is. There are narcoleptic mice, there are narcoleptic dogs, Emmanuel Mignot at Stanford, famous for that, and there are narcoleptic patients. And these systems have important descending pathways. Histamine also has descending pathways. In the paraventricular nucleus of the hypothalamus, parva cellularis, there we have oxytocin and vasopressin descending to the spinal cord and controlling the autonomic side of arousal. Obviously, when you're highly aroused, your breathing changes, your heart changes, and so forth. And finally, there are pathways in the preoptic area which come down and control autonomic arousal. Erection and ejaculation, for example, depend absolutely on this pathway. So we have ascending pathways for arousal and descending pathways for arousal. But I have to admit that I've fallen in love with neurons which could contribute to both ascending and descending paths for arousal. And these are giant neurons in the medullary reticular formation, which is just above the spinal cord. So if you look at my hand, you know where my spinal cord is, and you know where my cerebellum is right here, but just above the spinal cord in the medulla, in the lowest parts of the brain, there are these giant neurons, and they are properly called nucleus gigantocelularis. And what got me interested, there are many things, but to be brief, I'll just say that these are large neurons which are easy to deal with. And here's the axon coming out of that large neuron, and here's one branch of the axon descending toward the spinal cord, I believe, to support autonomic arousal. And here's the other branch of the axon, here's the branch right there. Here's the other branch of the axon going toward the midbrain reticular formation, which will then wake up the thalamus and the cortex. Here's another example of the same guy. Here's the Golgi stain showing the entire neuron. There's the cell body. The axon comes out, crosses the midline, bifurcates into the descending limb toward the spinal cord and the ascending limb toward the medulla. So this is nucleus gigantocelularis. I'll call it NGC for short. And I believe that these NGC neurons have the neuroanatomical, the neurophysiological, and the genomic properties to be, if to use a New York term, first responders. That is, when something is really alerting you, you have to do something fast because the bus is coming around the corner and you're about to die. I think that these neurons are the first guys to activate virtually every arousal system, sub-arousal system, as you have. Yes, please.
The first responder, you still have an operator, someone that tells them what to do. So what's driving these neurons?
Sensory input.
Straight, direct.
I think so, yeah. They have incredible dendritic trees. In the words of Ramon Moliner and Nauta, they're isodendritic. You take your first dendritic segment, it's a certain length. The second dendritic segment will be longer than the first, the third longer than the second, isodendritic. Go all over the place. And actually it's rather annoying because when I record from these neurons in slices, I've cut off all the inputs. And so I have the beauty of being able to do patch clamp recording, which I did on Tuesday. But I've denuded them of their inputs. And so as I go for precision, I'm losing their natural inputs. That's what drives them.
Okay, so but it's unprocessed sensory signals from the periphery.
Well, there is no such thing as an unprocessed sensory signal. The first stage of sensory processing is at the receptor. It's already a filter. And then there'll be another filter.
After that, from the periphery to these guys, nothing happens to the signal.
Not one hell of a lot.
Okay.
Right. And so a man, a neuroanatomist in Groningen in the Netherlands, Holstege, Holstege, would insist that yes, Don, you can emphasize these neurons and do precise work with them, but never think of them as acting alone. So I wasn't going to say this, but I think of these neurons as the evolutionary descendants from the Mautner cell of the fish. If you are a fish and you are just sitting there and something comes that disturbs you, it's the Mautner cell, which is so big that you can see it with your naked eye, which tells you to get out of there. I believe that these have matured in the mammalian brain from one Mautner neuron to a bunch of NGC neurons, and from just an escape response to alerting the entire CNS. Right. And if you want to see that argument spelled out with Don Faber, Mr. Mautner, it's in TINS of 2012.
Okay, great.
Yeah,
so these systems that arouse the brain themselves are regulated, presumably, apart from environmental input, by circadian and other regulatory.
And I'll talk a lot about that. Yeah. So Partha's point is that these neurons have to be regulated, and this arousal systems in general have to be regulated by circadian influences. And so you have the PER gene, you have the transcriptional feedback in circadian, which I can recommend. On my campus, the work of Michael Young, and the Brandeis campus, the work of Michael Rossbash would be the leaders, the genetic leaders in those respects.
And just a related question, are there any longer regulatory timescales, for example, related to depression or any other?
So the question is about longer periods of regulation. And I would be foolish to say no, that there aren't. For one thing, there's annual, right? And we have some breeders, I'll talk about sex later on. We have some breeders which are annual breeders. Usually, if you're engineering a reproductive system, you try to have it so that your babies are born under permissive conditions. So if you're a lamb, if you're a ram, you're going to try and fix it so that you're copulating, that after the gestation period, the baby will be born in a time when there's plenty of food around. And ethologists could give you plenty of other examples of that. Of course, we're freed from that, except during dark blackouts. We made a lot during electrical blackouts.
In the dark, that is.
In the dark, yes.
But another thing, just for clarification, how big is this population of neurons in the human brain?
So the question is, how many of these neurons are there? Well, in the human brain, I don't know. Let me think. Somebody in my lab could answer this. I think we're talking about not more than tens, but not tens of thousands. And so we're probably talking about 1,000 neurons, something like that.
Okay.
And I started out with the simplest, most parsimonious assumption that all NGC neurons are alike. That's obviously not true.
Right.
And some data that I won't talk about that, again, if they were doing what they were supposed to do after I headed for the plane, our most recent data would be Wednesday, where we have these guys firing just prior to cortical EEG changes. And it looks as though these are, and we can manipulate these neurons and manipulate cortical EEG. I will talk about that. But the reason I'm going into this is that not all NGC neurons do that. So even though I started out with the simplest assumption that they're all the same, they're just like an amplifier, clearly different NGC neurons have different relationships. And I'd like to know what those differences are, but even a year from now, I wouldn't know that. Yes, Nick?
So you don't think these are segmentally repeated down the spinal cord? If not, how are they recruiting sensory inputs from more caudal periphery sensory structures?
Up the spinal cord. These are not, as you're right, these are not repeated down the spinal cord. So that's why I said there's no such thing, especially at the level of the medullary reticular formation, as an unprocessed sensory signal. There will have been filtering, for instance, for a painful signal. When that painful signal stimulates the naked fiber, which comes to the layer 1 of the spinal cord and up to here, that's probably going to have a privileged and fast route. Yeah. So the signals will have been processed to some extent by the time they get here. Yeah. There are other interesting neuroanatomical takes on your question, but it would take me a while to talk about them. So let's talk about neuroanatomy. Nucleus gigantocellularis, we're not going to go through this in detail, these neurons project to all levels of the spinal cord bilaterally. That's the descending, I would think of it as autonomic arousal and arousal of the motor system. And in terms of ascending, all over the place, including, importantly, the midbrain reticular formation. If you get a stroke, or if you fall off your motorcycle and you damage this mechanically, medullary reticular formation, you are in a coma. That is trouble. That's how important the system is. And that's the reason that neurosurgeons don't go here. Any screwing around with NGC, you're likely to kill your patient. Here's a side point. In one electron micrograph, just one, we saw the nucleus gigantocellularis neuron. This is not an electron micrograph, up against the blood vessel, and there was no blood-brain barrier. I think most of you know that almost all of the brain, except for certain little windows, have a blood-brain barrier such that ugly things that are in your bloodstream cannot get into the brain. But this one electron micrograph told us, holy smoke, maybe these arousal cells are sensing things in the bloodstream. Could that be important for neuroimmune diseases? Could it be important for AIDS? And so we couldn't trust that we would ever get that electron micrograph again. So we started working at the light microscope level with immunocytochemistry. And here you see these extremely suggestive pictures. Here's the neuron in green. Here's the blood vessel, and this is tomato red stain in orange. Here's another one. Look, that's wound around in a sinuous fashion. Here too, here too, here too. And so all of this is suggestive. But at the light microscope level, there still could be an astrocytic glial cell between the blood vessel in orange and the nerve cell in green. And so what we then wanted to do was to inject stuff that should not be able to enter the brain. It should not be able to cross the blood-brain barrier. And so here we have, I think you can see a picture, a cross-section through the mouse medulla. And here's NGC in here. And here in the inset, you see that this neuron, this NGC neuron, has taken up the dye that does not cross the blood-brain barrier from the bloodstream. And it indicates to me that a small number of these arousal-related cells may actually be in communication with the bloodstream. Whether it's important or not, well, I think it probably is, but we have to get a lot more data. Just a side point. So much for neuroanatomical properties. What about electrophysiological properties? Do these cells have the properties expected of first responders, important neurons for fundamental generalized CNS arousal? And we had 5 criteria, and the answer is that over a period of 3 years and some excruciatingly difficult studies, Eugene Martin, he's published this in Journal of Neurophysiology, said yes, some of these cells are responsive to all sensory modalities. They habituate to repeated stimulation the way they should. They drive the cortical EEG. They drive the neck muscle EMG. And they're associated with the activation of behavior. In this case, it's a rat. If the rat's just standing there and the NGC cell is not firing at all, then the NGC cell fires, and within a couple of seconds, the animal's beginning to move. And I'll show you just some raw data about point number 1, responsive to all sensory modalities.
Quick question.
Yes, please.
This blood-brain barrier crossing presumably has important implications for drugs.
Yes. The point is drugs circulating in the bloodstream. Drugs that might not cross the blood-brain barrier could conceivably affect these neurons in the medullary reticular formation.
Has that been looked into, or
Excuse me?
has that been looked into? Is someone trying to exploit
No,
that?
no. This discovery, it's hard because we discovered it by accident, and nobody in the lab has any money to study it. And so right now it's like low-hanging fruit that I am not able to grab. Yeah. Okay, so about sensory modalities. This is the work of Eugene Martin in Journal of Neurophysiology, and I'm sorry about this. This is a 0 right here, and here's a 0, here's a 0. There's a 0, and there's a 0, which is the time at which he gave the stimulus. You see and here's 8 repetitions. You see, obviously, response to tactile. Olfactory is kind of surprising. I'll skip visual for a minute. Vestibular, great, and auditory, great. And the reason the auditory is complicated is because that's where we did our habituation. But the visual is the one I skipped. And you can see that, yes, there are trials where there was no spontaneous activity at all. This is a function of time. And we put the visual stimulus on here and you get a response. But sometimes you had high activity as he approached the rat in order to give the stimulus. And one ethologist, animal behaviorist, told me that he might have been comprising a looming stimulus. That Eugene Martin, the first author of this study, might have been viewed as a hawk or some other predator coming, and that the animal actually alerted. And this cell certainly was alert before he actually gave the visual stimulus. But I can't prove that. I just don't know.
But these stimuli, what were the stimuli exactly? Like the tactile stimulus?
The tactile stimulus was hair deflection. The question is, what is the stimulus? The tactile stimulus is a hair deflecting stimulus. That is just a puff of air, not a Von Frey hair, no. The olfactory stimulus is benzaldehyde. If you're recording from the olfactory bulb, benzaldehyde is the smell of almonds. So it's that German marzipan, that nice candy. Marzipan, yeah. And so the active ingredient of that is benzaldehyde, and the virtue is that it makes every mitral cell in the olfactory bulb go crazy, but it does not have a trigeminal component. Lots of times your olfactory stimuli are stimulating the trigeminal area of the nose as well as the olfactory. The visual was just a light, the vestibular was rocking the platform, and the auditory was a click, having many, many frequencies represented.
So the visual was just a flash of light?
It was just a flash of light. Yeah, it's kind of stupid giving a visual stimulus to a mouse or a rat because they're not really visual animals, but we had to for the sake of completeness.
But they're derived from very visual rodents.
Yes.
In nature, you have species of mouse that are highly visual.
Nick makes the point that some mice are highly visual, and by the way, it's not that mice can't see, and it's just that their vision doesn't play as large a portion of their life as it does ours. So another way of studying the physiological properties of these systems, up till now I've just been measuring the electrical activity, but another way is to mess around with these neurons using electrical tools or chemical tools to say, if you change the activity of these neurons, do you get changes in arousal? I'd better speed up. And so here we're electrically stimulating gigantocellularus. Here you have a sleepy electroencephalograph, EEG, which would be large amplitude waves and low frequency. And when you stimulate, and this is where we missed NGC with our electrode. We did the histology afterwards. We missed it. But here we were on target, and when we stimulated, we got the alert EEG, which would be low amplitude, high frequency. And this is a perfect control, because when we did the experiment, we didn't actually know that we had missed. We didn't know until later. So this was an unintentional control. So there we're monkeying with these neurons electrically. Now let's mess with them electrophysiologically. Here we have the cannula tracks down into NGC, gigantocellularus. Here we had the anesthetic hooked to a fluorescent molecule to prove to you that the anesthetic that we were injecting into NGC was not going over the entire brain. It was just in NGC. And here's the effect. Electrically, we're recording EEG here. Here's an awake EEG. Here's a sleepy EEG after injection of anesthetic just into NGC. And here's the recovery. Behaviorally, here's low signs of anesthesiology. Here's high anesthetized, and here's low anesthetized. So electrically and behaviorally, if we monkey with NGC, we can manipulate overall arousal. Now let's do the opposite. Instead of anesthetizing NGC, we'll put in a GABA receptor blocker which fights against the anesthesia. And here you have the anesthetized EEG. Here you have the awake EEG after we have blocked GABA receptors in NGC only. And here we have the sleepy EEG again. Here's the recovery. Behaviorally as well. This is respiratory rate. The respiratory rate goes up when we put the blocker just into NGC. And here's the recovery. And here is the latency to movement, lower latency to movement after we block GABA receptors. And the most primitive experiment was actually the first one. In this case, we're looking at response to foot pinch. One of the things that you're anesthetized, obviously, for during surgery is to prevent pain. And the animal's not responding to pain. After we put the GABA receptor blocker into NGC only, nucleus gigantocellularis, we get some response to pain. Then it wears off, and then the animal eventually will recover. So physiologically, NGC can do the job. And now genomically, you'll recognize this as RT-PCR, a reverse transcriptase polymerase chain reaction. And here, orexin, remember the peptide that I talked about before with narcolepsy, orexin 1 and orexin 2 receptors are expressed in this individual neuron. This is an individual NGC neuron where we went into a slice and we sucked out the messenger RNA. Orexin 1 and orexin 2 receptors are expressed. And then for lowering arousal, opioid receptors, like heroin, we have delta opioid receptor and kappa opioid receptor and mu opioid receptor all crossing this threshold. That is all showing detectable messenger RNA in this particular neuron. We've never followed this up. All we can say is now we know that anatomically, physiologically, and from this slide, genomically, these neurons seem to be able to do the job to regulate arousal. The final slide of this series is that if you are wanting to respond very rapidly to a stimulus, wouldn't it be nice if once one NGC neuron is stimulated, that the electrical excitation spreads rapidly? And one way of doing that is to have gap junctions. And you may know that if you have two neurons, if you look at my hands, which are next to each other, if their gap junctions are lined up next to each other, such that you get a full gap junction, then electrically charged particles can go from cell number 1 to cell number 2. Now, these gap junctions are composed of hexamers of connexin 36 molecules. And what this slide shows is a transgenic mouse in which, from the connexin 36 promoter, the green fluorescent protein is expressed. And then in NGC, connexin 36 is, well, here at low power, but here at high power, and this is the most exciting part, where two neurons which are expressing green fluorescent protein from the connexin 36 promoter, which means that they also should be expressing connexin 36, are next to each other. Now that's not actually a proof of a gap junction, because it could be that they're expressing the messenger RNA, but the protein isn't getting made. Or maybe the protein is getting made, but it's not lining up properly. But it shows that these cells have the capacity, at least, to express the genes which would make a gap junction. Therefore, raising the possibility, but we haven't proven this, that the activity of these giant cells in the reticular formation may be able to be amplified readily.
If these were homologs of the mouth of the cell, you might expect them to be highly equipped with gap junctions.
I didn't know that. So Nick said that if these are like the Mautner cells, that you would expect them to have gap junctions. I actually didn't know that. So that's encouraging. The discouraging part is doing the electrophysiology with paired cells in order to show that the gap junctions are really operating at high efficiency. Partha?
Two questions. One is there a reason why it is in contact with the blood? Namely, is there some blood-borne molecule that the body is using to influence it? And the second question is, did you say it projects to cortex? I missed that.
Midline thalamus. Not all the way to cortex, midline thalamus. So the second question is about the projection, midline thalamus. The first question was about that blood-brain barrier question. And so yes, a nice fantasy for a person who is devoted to these neurons, at least this Friday morning, would be that drugs that have something to do with arousal, like drugs of abuse, will affect one of these neurons, and both through synaptic projections and through gap junctions, general arousal is affected.
But is the body using it? Is there a reason why it is that way, you think?
So the question is, what about the evolutionary advantage? In other words, is the body using this mechanism? And the answer is, I don't know.
No, I think part of means something else. I think, in some sense, if these NGC neurons are directly sampling the bloodstream, you could imagine that the body releases substances that directly talks to these neurons.
Yes.
And that then drives central arousal
Mhm.
from the periphery. I think this is the point.
Yeah, but
Yeah.
he's saying he doesn't know. It's
The,
a hypothesis.
the answer is that it's a nice thought that the body is using this device in order to regulate, because one of the, actually in a sense, one thing I do for a living is to talk about relationships between the rest of the body and the brain. That's what all our hormone studies are about, is how does the brain stay in touch with the body. And Partha's point is that yes, this would be a good mechanism for doing it, but the reason I was shy about it is that we don't actually have the data to prove that it does. Nick?
For a herbivore, this would be a very efficient system, an early warning system, if in ingesting some toxic or opiate substance, I think.
Yeah, so the point was that this would be a wonderful warning system. And in fact, a man named Paul Rozin, R-O-Z-I-N, at the University of Pennsylvania, has talked about rats being able to detect something bad in their food, and they avoid that food forever. What is this called? Taste aversion. If you have the laws of learning, which all of us have and which rats have, and they have certain quantitative properties and temporal properties, taste aversion, by which the animal does exactly what you just said, Nick, has entirely different properties. It's so important that taste aversion lasts forever. I got sick after I ate knockwurst in Cambridge, Massachusetts, when I was 20 years old. I've never had a knockwurst since.
Another question.
Yeah.
Don't worry too much about time, because we'll only have 2 speakers in the morning. Lunch is at 2:30.
Okay.
That'll be fun. Oh, sorry, 2. We lost half an hour then.
Yeah.
So the question is, what you showed with the pharmacological study is that the GABA receptors are playing quite a role in regulating these neurons' responses. So on the other hand, you say, well, they're directly driven by the sensory periphery.
Yes.
But now there's actually an additional step in this game,
Yes.
which is some form of inhibitory control over the response of these neurons.
Yep.
So where is that coming from? Is that
I would like,
local
so the
control?
question is, in addition to these sensory drives on these neurons, what about this GABA? Because we did that pharmacological study 2 or 3 slides ago. And the answer is almost certainly, but we haven't proven this, that there are local neurons, which are tiny neurons, which are GABA neurons. We know that they're there. But the idea, in answer to your question, would be that they are regulating the activity of these neurons. And that when we went in with our pharmacology, we were mimicking the action of those local neurons.
Okay.
And that's one of the major questions in neuroscience, is that lots of our techniques for students here are well adapted to looking either at individual neurons or at neurons in the conglomerate, like the entire brain. But our techniques for dealing with populations of neurons which form functional units are not so great. And like you could go back all the way to 1964, and there was a great neurophysiologist named Stephen Kuffler, and there's a paper by Orkand and Kuffler in 1964, which showed in leech the glial cells of the packet. And what that meant was that there was a functional unit which was defined not by neurons but by a glial cell, and they all worked together. If I were to have every dream answered in answer to your question, it would be that there's a packet of cells in the medullary reticular formation. Here a packet, there a packet, there a packet, there a packet. And that each packet has one or more giant cells with a bunch of little guys nearby. And those guys that do, I think it's Barabasi? No, or have I mixed them up? But they talk about local connections, like Bill Clinton is NGC, and all the rest of us are like tiny little cells. And they talk about cells talking to other cells and the advantage of that. You could see, it has been argued that there are advantages to having some cells have very small number of connections and local business, and other cells be talking over the long distance. People have made that argument. I love to read them, but in my case, I certainly haven't proven any of them.
And how about intrinsic dynamics of these neurons? Are they intrinsically active, or do they really depend on external input?
That's what I was doing on Tuesday, is trying to find out the rules by which they operate. And what I'm really looking for is biophysical properties that would make them as special as I would like them to be. But I haven't proven that yet. I've been working on it quite a while. And so far, I haven't proven any magical X factor in these neurons, like a channel that's there.
Because in the physiology you showed, they seem to be very much sensory driven.
Yes.
With one exception for the visual stimulus.
Yep.
So this is roughly their average physiological signature would be like that.
Yep. So the question was about the rules by which these cells operate. I was hoping that I would find an X factor that would account for their power. But so far, the disappointingly mundane conclusion is that these cells are dominated by their dendritic inputs.
Can I make a comment?
Yes, Nick.
Comment. I mean, in the mouse system, or for any escape, giant neuron that's involved in escape, you need to just keep that neuron silent most of the time.
Mm-hmm.
Otherwise, it loses its magic. Right?
Yep.
But you expect, if this is a homologue of the mouse cell system, to have very, very tightly coupled GABAergic pathways to inhibit these cells. After they've done that they perform their alerting function.
So the point is that it's important for a cell that's so powerful to keep it quiet when it shouldn't be active. Nick's right, and our data would say that yes, these cells are quiet most of the time, and we believe but have not proven that the local GABA neurons do the job.
Well, I'm not sure if this holds actually this comparison, because for the Mauchner cell, you want to trigger a very specific behavioral response, right? Like escape. And you're proposing that these neurons are driving more general arousal within the context of which the system operates for, let's say, hours on end.
Yes.
You still need to keep it quiet when you still need to have a circuit that is responding to, that is not responding the whole time. Otherwise, it loses its function as an alert system. The same is true for the Mauchner cell.
But the Mauchner cell has to be, let's say, excessively transient. Well, in this case, you want to open that window of response a bit more. This is the point I'm posing.
I think I agree with both of you. That yes, it's important to keep the cells quiet so that when they do fire, it really means something. On the other hand, it's definitely true that if the Mauchner cell is devoted to escape, I am proposing, just as you said, that these cells are at the basis of every motivated response there is. They don't account for everything. They don't account for the hunger when you don't have food, or the thirst when you don't have water, but they account for they give the juice to the behavior.
Do you know the subtypes of the local GABAergic neurons?
Excuse me?
Do you know the subtype of the local GABAergic
I wish I did. Question is, what do we know about the subtypes of those GABA neurons? I really wish we did. If we had more grant money, as you said a couple days ago, funding agencies permitting.
It might be something I could shed light on, because we are now imaging all the subclasses of the GABAergic neurons.
Yes.
And these cells, they are really visible without any staining. The NGC cells. So one might be able to tell just by looking.
Yeah. And we're trying to collaborate with Pavel Ostin on your campus, but boy, is he slow. Anyway, so we don't know as much as we want, especially about the GABA cells. And I feel comfortable dealing with the big cells, but I fully realize that they're not the entire story.
But on the GABA receptor side, can you say something there?
GABA A receptors are the main story.
Okay.
But I think Partha has something more in mind, because among GABA A receptors there are still further specializations. Well, I think everybody's had a chance to read this. I mean, we really want better molecular characterization, as you gentlemen are pointing out, not only of the big cells, but also of the little cells, likely GABA cells, and what are they really sensing. So one of the reasons for having the guts or the foolishness to deal with this global concept of generalized arousal is that we can generate tremendous amounts of quantitative data very rapidly without working. The mouse does all the work. And we come to this first of two quantitative statements. Here's the first one, is that several workers, and this applied physicist Barabasi is the most famous among them, I think, have published power law statistics, which I'll explain, of human behaviors, like responses to email latencies is one example. How long does it take you to respond to email? And if you do that for hundreds of thousands of cases, you see power laws, which I'll explain in a minute, in complex settings. Also fish which are looking for food, show a lot of power law behavior. And the response, in all of these cases, responses to the environment are paramount. And here's my major point of the next few slides. We've discovered power law behavior of mice under circumstances where we're looking at the properties of an intrinsic generator. There's nothing changing in the environment, as you will see. And it's so important that we work with the physicist Jayanth Banavar, who's just moved from Penn State to University of Maryland, and with the Italian mathematician Amos Maritain, who used to be in Trieste, and he just moved to Padova. Everybody moved, except me. what Alex Prote, this anesthesiologist in my lab, is doing is he's showing that locomotor activity is structured in discrete bursts, here in days, here in hours, here in smaller number of hours, here in minutes, and here in seconds. These bursts of activity present at multiple time scales, as I just said, with temporal structure of activity appearing to be self-similar. And here's the point, is that when you have a straight line on a log-log plot, what you're dealing with is a power law. So there's the exponent of the power law, and this is the equation. And what we have in animals which are totally separated from any external stimuli is power law behavior over 3 orders of magnitude. Here's the 24-hour rhythm right here. Power law over 3 orders of magnitude of this intrinsic motor activity.
How do you define interlocomotion interval?
It's movement. We're measuring movement.
That's a very imprecise definition for someone as precise as
Well, the beams are spaced 1 centimeter apart. And in order to trigger it, the animal has to do 2 in a row, which means that he not only just moved his head like that, but he also went from here over to here. And there's nothing complicated about that.
That might be one of the, we can have this discussion later, but if you look at the work of Ilan Golani, for example,
Yes.
he carefully characterizes rodent locomotion in an open field arena.
Yeah.
One has to actually, there's some complexity to characterizing when the organism is at rest and when it's moving.
Yeah.
We can make this discussion offline.
Well, actually, I will quote that in a minute because you're right. His name should come up. You can make it complicated, but my interest is in making it simple.
Yes, but when people present these power laws, often they're artifacts of data analysis. This is the reason why I'm bringing it up.
I'll show a couple of slides, and we'll say after that, am I likely to be subject to some artifact? One of the reasons I'm here is to look for artifacts.
So the restoration would not be Poissonian, you're saying? It is a power law, because if it was Poissonian, you would, let's see, what would you have there? I'm not quite sure what it would look like.
No, this should be the equation. Our exponent is 0.7. And so what is required is that the animal break a beam and then break another beam in that direction. And I'm looking for simplicity. Now, Partha was referring to his collaborator, who's a brilliant behavioral neuroscientist in Tel Aviv named Ilan Golani. And just to be educational about this, but Ilan Golani, the first paper was in Biological Cybernetics, 1994. And there's been a series of publications since then, and a publication from Ilan Golani in PNAS of 2010. And if you put a mouse, I saw this on a table, and it does nothing special, on a table. But usually what he does is he puts the mouse in an arena, like the gladiators, in an arena, and he has one little mark in the arena which the mouse is willing to call home, even though it's not really the mouse's home. He puts the mouse there, and the first time the mouse circumlocutes, he goes around a little circle, and he pauses a lot, and he goes at a low rate. Okay, we're still alive. We're back home. The next time we're going to make a larger diameter faster, next time larger, and eventually he's circumlocuting the entire arena. And when I was visiting his lab, he showed me that. There's a bunch of us were just sitting around. He brings in a mouse, and he puts it on one end of a table about half the size of this, and the mouse does exactly what I just said. So it's a tremendous lawfulness with a greater degree of complexity and probably some good ethological verification of why that should be, that the mouse does. But I'm looking for something lower. I'm not looking for something more fundamental. I'm not looking for all of the ethological characteristics of a mouse. I'm looking for what wakes up the entire system.
Well, what happens in cases when one finds broad distributions is that there's often a mixture, if you want, of Gaussians.
Yes.
So that, and then you can only find it if you look more closely at your data. But we can discuss this offline because there's a lot of history.
In this case, you cannot really distinguish translations from rotations, and so you have mixtures of these, and these will definitely have an impact on the distribution of your data.
A paper which will be submitted real soon with the mathematician named Dan Keenan, a professor at University of Virginia, who's a visiting professor in my lab, did manage to distinguish it. And just yesterday I was reading the draft and trying to understand. And now he says he's distinguished this from simple rotation. If the animal stays in a very tight circle.
Look, we can first look at the data and then hassle you about it.
Yeah. But that is,
But there are issues.
that is the question, because, for example, if the animal had damage to one side of its basal ganglia, which we heard about from Nick, the animal might make tight rotations, which is not that would be a disease or whatever. I'm trying to talk about normal behavior.
No, sure, but all I'm saying is that the mouse is not always just translating in a straight way through
Absolutely
the
not.
regions.
Yeah.
And it's also not always making tight circles, so there's whole distribution in between.
Yeah.
And this can be an example of the kind of artifacts and data sampling that the part is alluding to.
We have that covered like a tent. And if you email me, I'll send you the next draft of the manuscript. This draft is written in mathematics. It's not written in English. And so, yeah. Okay, so we did want to know how many mistakes have we made in discovering this power law. One thing could be that the way you get power law statistics is by the animal spending a tremendous amount of time in one place and very small amounts of time in lots of places. But that's not necessarily the only way that we generate power law, because here we have data coming from an individual place, just one place, and again we get power law statistics, a straight line and a log-log plot. Another question is, what about light and dark? We get better power law behavior in the dark than we do in the light. The light is the dashed line. Remember, these are nocturnal animals, and here's the animal behaving in the dark. Another thing is that we could have messed ourselves up by applying statistics incorrectly. And so what we did, this is Kolmogorov-Smirnov, and what we did, what Alex Procht did, is to generate 10,000 artificial power law data sets. And he did statistics on them. And yes, indeed, they do show power law statistics. And here, in our own mouse data, we have either 6 or 7 mice piled up, and here's another mouse here with a p smaller than 0.05, whereas out in the light, not consistent with power law, we have a smaller Kolmogorov-Smirnov estimation. So it looks as though we're doing the statistics correctly, because our actual mouse data, 6 or 7 mice right here and another mouse there in the dark, are showing p smaller than 0.05 as expected. So up until this point, we're just doing animal behavior, and we're doing a lot of calculations of the sort that I've shown over the last 2 or 3 slides. But then something surprising happened. The physicist Jayanth Banavar, who, as I said, moved over to the University of Maryland, and Amos Maritan, who's now moved to the University of Padova, took another look at the data. And so yes, here's what they would say, and I hope for reactions to this. We have, the way we do this is we have nerve cell electrical activity generating behavior, and it works on an interval of milliseconds, time scales of milliseconds. But the fluctuations of behavioral arousal, which I'm so devoted to, occur in the range of minutes to hours. Their derivation, their mathematical derivation, which I trust, but I can't do it myself, and you see it in the appendix of the paper, which just appeared in PNAS, is that with this scope of timescales, power laws emerge inevitably. In other words, it's really, they would say, an inevitable consequence of the fact that you've got dynamics in milliseconds and behavioral dynamics in minutes to hours, that you're going to get a power law no matter what.
That statement simply can't be true, because one can make a counter model where you have, specific distributions of these behavioral arousals that are not power law distributed.
Whereas what Jayanth and Amos would say, I'll speak for them for a moment, is that not only can be true, it has to be true. And our worry about this, Partha, was not that we made a mistake. Our worry was that we had uncovered a tautology. And that is still one of the reasons I'm here, is to explore that. Did I really, working with these guys, did I really now witness a discovery about the properties of arousal systems, or did I restate a tautology in the fanciest possible way? That's the real worry. And so the positive way of looking at this, and again, it's two-faced. Is it a discovery, or is it a restatement of the obvious? Is that from this thinking, power law distribution about CNS arousal derives automatically from the intrinsic dynamics of neuronal activity and does not necessarily depend upon the environment. It can depend upon the environment when we answer our emails, as half of you are doing. We may be generating power law statistics, And what's another example? The fish is another example, responding to food, generating power law statistics. So there can be environmental causes, but our data suggests that that doesn't have to be. So what I would invite you to do?
There are examples of this kind of thing going on in the heart, for example, where people have fit power laws up the wazoo, but then if you were to speak with John Doyle, he would explain to you that environmental inputs can indeed drive these broad fluctuations. So I do not actually, I'm very skeptical of what you're saying. I do believe that you have observed these power law histograms, but I suspect there may be explanations which are not consistent with an intrinsic dynamics that has a broad distribution of time scales.
It doesn't depend necessarily on intrinsic. All I'm trying to say is intrinsic can generate it. So I'll answer in two ways to what Partha has said, and I'll also repeat what you said. That on the one hand, we can be skeptical, and I would invite you to look at the supplementary data, the actual mathematical derivation, and see if you can find an error, but you won't.
I'm interested more in the data than
yeah, but the second side of what Partha said is that a control engineer named John Doyle, who is a professor of engineering at Caltech, has dealt with these questions with the dynamics of the heart, and that he's achieved certain statements about outside influences on heart cells beating, I think is probably what it is. And so I would invite you to look at that, because he certainly is a well-known control engineer who deals with biology.
Another worry that I would have, because we look at locomotor activity,
yeah.
right?
Yes.
And locomotor activity is often related to the intrinsic dynamics of the inferior olive, which is then seen as a motor clock, if you want.
Yes, yeah.
Right? And we know from the inferior olive that indeed it has an intrinsic dynamics that
yes.
can run at about 1 to 10 hertz.
Yeah.
And that's about it.
This is Rodolfo Llenas?
Well, not only Rodolfo would say that, also Mike Malk and others.
Many people.
And then they're sort of, what they don't agree on is the exact frequency. That's where but it will be in the range of 1 to 10 hertz. But that is pretty much like a clock-like system, right? So if your operant definition that you're analyzing is locomotor behavior, and we know that in itself is clocked in a rather fixed range of 1 to 10 hertz, then these power law properties might indeed more coming from other perturbations on the system than being intrinsically driven. Look, I'm inventing a story here, but that would be an alternative to your point 4 here, because you really want it to be generated intrinsically.
Well, the inferior olive is in the brain.
You know, but I thought you were more interested in your arousal system.
I am. Yeah, I am.
So and the inferior olive is a pretty tightly closed system.
I'm most interested in NGC, but when I said intrinsic dynamics of neuronal activity, certainly another cell group which is in the medulla called the inferior olive certainly could be playing a part in this ball game. Yes.
No, what I'm saying, they're so tightly clocked that it would be hard for me to see how this can give you your power law. Yeah,
I haven't thought about that, but yes, I'm not sure how that would work out. I'm not sure either neuroanatomically or quantitatively how that would work, but I agree that I should think about it. Yeah.
So the reason I'm being so skeptical is I've looked at locomotor activity myself in fruit fly more so than in rodent, and this is largely unpublished work. There was one paper, but I saw no evidence of power law distribution, but I did see a distribution of time scales. What you find is there are different episodes. Fly likes to linger, likes to walk. Has different lengths of walking and so on and so forth. You can find a fairly parsimonious description of what's going on by breaking the behavior up into states. Each state has got some distribution and so on and so forth. If you were to lump all of it together, then you might start getting broad distributions. But it doesn't have any profound kind of origin, I think. It's simply that there's a distribution of different things happening, each interpretable with a different, if you want, behavior associated with it.
Right.
So what Partha is saying, without referring to details of unpublished data, is that it could be that the power law it derives in part from large numbers of disparate activities which have separate biological meanings and which have different temporal dynamics.
And not that many of them. In fact, 2 or 3 will do.
So if we word it that way, we take some of the mystery and the magic out of it. But I still like this statement because it puts the mystery and the magic back in.
But why is that nice?
Hmm?
Why is that likable?
Oh, because it's universal. I'm going for stuff which is universal as possible. And it may be grabbing for too much. I think Parther would say, in grabbing, I'm ignoring a lot, which I probably am.
That's really a challenge, right?
Yeah, it is a good challenge. So now, one of the reasons I like this is that this same physicist, Jayant Banavar, and I talked a few years ago about phase transitions. And sometimes power law dynamics come in systems which are close to a phase transition. And in a somewhat playful manner, we made an analogy to a liquid crystal, where at a slightly higher temperature, you have disordered molecules, and at a slightly lower temperature, completely ordered molecules. And we argued this about 5 years ago. And what we were saying in that was that perhaps we don't believe that the brain operates in a linear fashion. And we were playfully suggesting that the animal which is quiescent, the arousal system which isn't working, and it's just quiet, is very close to a phase transition, and it's operating in a chaotic dynamic. That is, it's taking advantage of the amplification power and the rapidity of nonlinear systems. And then, when the animal actually begins to move, it goes across the transition into a period of ordered movement. And that it could be that the power law dynamics come from being close to this phase transition. But that's just a happy thought. That's one of the reasons that we're excited about it. So what we're doing now is really, we want to study animals in dark, so we're really looking at the circadian clock. Up until now, we've been working in light and trained systems. We're interested in genetic knockouts and how they influence it, especially for GABA receptors. Obviously, we're interested in anesthesia. That's what we're doing right now. Also, another thing that I'm going to show you that we're in the middle of now is generating dynamic fingerprints of behavior, which you could do for any animal. We happen to do for mice. And then, as I've said, Jayant and Amos with that mathematical derivation lead to a reconceptualization of power law applications to behavior, which may or may not be interesting.
So, question to that. These behavioral fingerprints.
Yes.
Like people like Peter Wolf and Hans-Peter Lipp have come up with something like 24 unique segments in the water maze in mice.
Mm-hmm.
Is that a plausible way to think about behavioral fingerprints, or would you think about smaller numbers of behavioral fingerprints?
In the next 5 or 6 slides, I'll show you what I mean by behavioral fingerprints. But what you were talking about was Hans-Peter Lipp, who in a somewhat richer environment has talked about behavioral fingerprints. And so far I've tried to avoid that, actually, partly because it's already been done, but partly because I'm looking for the most elementary, fundamental, vanilla flavor quantification of behavior that I possibly can. I'm going for deeper than deep. So these are things that we're in the middle of right now, the next 4 or 5 slides, and we'll see the part that I barely understand. So Alex Prote has gotten interested in the frequency representations of these behaviors I've been showing you. We're still talking about this generalized arousal assay. The animal's untouched by human hands. There's no new stimuli coming in at all. And if we have the typical 1/f distribution. Here's frequency. Here's power, probability. And what Alex has gotten interested in, if the circadian rhythms are over here, he's gotten interested in the ultradian rhythms. These would be rhythms which have periods which are much shorter than 24 hours. They're more rapid. Ultradian would be the word. He's gotten interested in them. Now, we don't believe that behavior is stationary. The quantitative features of the behavior are not going to be constant over time. And therefore, what Alex Prote has done is to apply a transformation, which is a standard wavelet transformation, what happens to be new to me, which gives us both time and frequency information, both time information and frequency information. And Alex is trying to work this to see how far we can get. And we're in the middle of it right now. And what we have here is time on the x-axis and frequency on the, time on the x-axis, and frequency on the y-axis. And here's the 24-hour rhythm right here, and here are the ultradian rhythms right here. And he is especially, this is a heat map, where a greater interaction will mean a darker color. And what he's interested is in the interaction of different rhythms of behavior with each other. And you can think of examples in your daily life where you have rhythms of behavior interacting with each other, except in this case, it's just a mouse, and it's in a faceless environment, not doing anything at all special. And so an example of this is how would these behaviors interact with each other. And so what he's got here is one of the ultradian rhythms, shown right here, and he wants to look at the power of this ultradian rhythm, this more rapid rhythm, as a function of the phase of the circadian oscillator. In this particular example worked out very nicely. At one particular phase of the circadian oscillator, this ultradian rhythm is very powerful, and other times the circadian oscillator, it's very simple. Now you could say, gee, that's not very complicated. I mean, why bother doing that? And I'll show you why in a minute. We don't need this. It's to generate what you might call a dynamic fingerprint of behavior. And this could be applied to any vertebrate animal. This could be applied to humans by Homeland Security. It could be applied to any vertebrate, as I said. And what this plot is a measure of rhythmic behaviors, components of behavior, interacting with each other. Here you have the carrier frequency. Here you have the signal frequency. And here and again, it's a heat map. High modulation of one frequency by another will be in orange, and no modulation will be in black. And so here you have, on the carrier frequency, circadian very powerfully influencing a whole bunch of ultradian behaviors. But you also have an ultradian oscillator or oscillators, maybe plural, over here. So this is the kind of map that we want to get. And I'll show you why. Because then what we do is we apply this to our populations of animals which have been bred. Remember, about 3 hours ago, I was talking about breeding for arousal. And we are going from, we started out with a very genetically heterogeneous population, and from generation to generation to generation, we applied the operational definition of arousal, as I told you, and we have sensory responses, motor responses, and fear, giving a high arousal line and a low arousal line, and they give different dynamic fingerprints. So low arousal progeny are up here, high arousal progeny down here, and take a look at this interaction here and here and here and here, but we didn't see it here or here, and not so strong here and not so strong here. For example, if you compare this to this. So we think that some of these interactions between rhythmic behaviors have something to do. They either generate part of arousal or they're due to part of arousal. We're not sure which. All we know is that they seem to be different between low arousal bred animals and animals which are bred for high arousal. Now that's something that we want to analyze a lot. A somewhat amusing thing is that females have a 4-day rhythm. For the ovulatory cycle, like humans it's 28 days or 30 days. In a mouse, it's 4 days or 5 days. And we've discovered the traces of female rhythms in mice this way. Now we have it in so small a number of animals that I'm not showing it to you, but we're beginning to generate the possibility that in the male mouse brain, there's the ghost memory of a female rhythm, a 4 or 5 day rhythm. And it's not due to the females because there's no females around. It's the male mouse by himself. Whether that means anything that's other than a slightly amusing thing, I don't really know.
But doesn't it look counterintuitive? Because your low arousal progeny, which sort of is going out in this environment less frequently,
Yep.
doing less, and the measurement you're analyzing is how often they break those beams.
Absolutely.
Seem to show more rhythm.
So the question is the counterintuitive. These guys have interactions that these guys don't, as far as we can tell so far. Remember, we're just in the middle of this, and these are the low arousal. So what's going on? Why do the low arousal have these interactions? Now, I could give long, verbose rationalizations of how that could turn out to be, and I could give analogies, but I'd really be just spinning fairy tales. And so rather than doing that, I'll simply say that we're intrigued by it, and it could be that nobody else will be intrigued by it, and maybe a year from now or 2 years from now, we'll actually be able to say how this could come about.
Sure, but can you tell me what the behavioral pattern is behind this?
We don't know.
Okay.
We don't know. I take comfort, and the question has been, isn't this counterintuitive, I think was I grew up with the notion that paradoxes are good. That when a scientist hits a paradox, that means something important has happened.
Sure,
And so I believe that something important is going on, but I don't have a clue as to what Partha has a clue.
No, I was just asking that what I found interesting from what you've said here is not the scale stuff, but the fact that there are these ultradian rhythms. What
Yeah.
are the frequencies of those rhythms?
There's a bunch of them.
And what are they?
I'd have to go back a few slides, which I don't think I'd better do. But there were 5 or 6 peaks.
And do you know what they correspond to in
Alex knows.
Okay. Maybe you can, if it's a real rhythm, then you can ask what is the mouse doing? Is it, periods of quiescence and running, or
is it minutes or hours? Do you remember?
Those were in minutes.
Okay.
Yeah, and remember, the animal is just in this cage all by itself. Nothing's happening, so it's not responding to anything which is changing. It's generating it itself, and why it's generating it in those ways. So Partha's point was those peaks in the 1/f diagram, which I showed a few slides ago, they do correspond to certain rhythms, and that probably means something, but we don't have the faintest idea of what it means.
worth looking for that?
So we couldn't see the animals because they're isolated from everything, and now we're buying some new equipment. Actually, we're convincing Stolting to give us the equipment for free. We're going to beta test some equipment where we can see the animal, actually, and then we may be able to answer your question, if indeed they give us this equipment for free.
It's just the frequencies might give you a hint.
Oh, yes. No, I agree, absolutely. So I shouldn't say conclusion. Our present thinking about this kind of behavior is that the wavelet analysis of behavior may indeed be a useful tool. We've had previous conversations, and he has his questions about it, his skeptical questions. We think that behavioral oscillations occur simultaneously at different frequencies and are coupled in non-trivial ways. We hope non-trivial. And the analysis of this coupling may prove useful in discovering subtle behavioral differences in rhythmic activity, especially if we can find out exactly what the animals are doing. So now I'd like to whip to the second part of the talk. The second and the third are very brief. It turns out that I'm talking a lot about what we're doing right now and it's not finished. But now I can talk very briefly about things that took approximately 30 years to do. And so I'm quoting this famous Caltech professor, David Anderson, in Nature, where he says a central problem in neuroscience is to understand how instinctive behaviors are encoded in the brain. So in the search for rigor, as this first week of the meeting of the school is supposed to be about, one way of doing it is to do behaviors which you can mathematize, if you'll permit me that word. But another way of doing it is to actually choose a behavior, go to the other end of the spectrum, choose a behavior which is so simple that you can bring it down to very strict physical analysis, which is what we've done. So what we've got is a chain of behaviors, courtship followed by copulation, followed by maternal behavior, which is essential for reproduction by female mammals. And what we've determined is that an individual gene, estrogen receptor alpha, expressed in specific ventromedial hypothalamic neurons, essential for copulation by the female. And by the way, it's also expressed in preoptic area neurons, and it's essential for maternal behavior. So here you have 2 parts that are essential for female reproduction in a mammal, the copulation and the maternal behavior. And so here's the behavior. The animal does not even have to locomote. It's called lordosis behavior. If the female does not want to mate, any female with 4 feet, if it doesn't want to mate, it will stay like this with its rump down, or it might kick the male or bite the male. But if it wants to mate, under the influence of estrogen and progesterone, it goes into a dorsiflexed posture. This would be concave up, which permits the male to fertilize. And if the female doesn't do this lordosis behavior, then reproduction will not occur. It doesn't even require locomotion. That's how we could figure it out. And so what we know from the next 3 slides I'm going to show you is that we know how steroid hormones coordinate brain function with the rest of the body to ensure reproduction appropriate to the environment. So this is just like what Partha said before, where we got stuff that's circulating in the blood. In this case, it's estrogen and progesterone from the female's ovary affecting the brain. And we understand the mechanisms from receptor chemistry, which is measured in angstroms, through seasonality, where the physical parameters are measured in light years. So if you divide this physical measure by this physical measure, it's 10 to the 24th. And so we really have this figured out. And the proof comes in 3 steps. First is there's the hormone receptors. The hormones are circulating in the blood, and we're looking at a rat brain from the left side, and every little black dot is a nerve cell which binds radioactive estradiol, estrogen. And it's a so-called limbic hypothalamic system. That would be the limbic forebrain, like the hippocampus and the amygdala. Here's the amygdala right here. There's the hippocampus. And then hypothalamus, which I think you know. And we discovered it in rodents, but it's actually universal among all vertebrates. From the fish to the philosopher. From the fish to the philosopher, this system operates, especially if the philosopher is a woman. And the most important part of this is the ventromedial nucleus of the hypothalamus, right, that little VM right there. And the reason that's important is step 2 of the proof, because here's the ventromedial nucleus of the hypothalamus, and it's one of 3 anchors for this circuit. So this is the first circuit for a behavior in any vertebrate animal, this lordosis behavior. And the way of discovery was to go from 3 anchors. One anchor is the hormone-binding neurons here in the hypothalamus. The second anchor is the sensory stimuli on the skin of the female. And the third anchor would be the muscles and therefore the motor neurons which generate the behavior. And so what we did was we worked our way in from the sensory side. We worked our way backwards up the motor side until we found the estrogen-dependent signal coming out of the hypothalamus. And to make a long story short, the hormone-binding neurons, and this is true of any vertebrate animal, the hormone-binding neurons send a signal back to the midbrain, and the midbrain governs a sensory midbrain, a spinal midbrain spinal loop, which generates this behavior. So that's step 2. At that point, we got very lucky. These proteins, estrogen receptor proteins, in the hypothalamus turned out to be ligand-activated transcription factors. And so if you remember back to your molecular biology, you'll remember that transcription factors are proteins that bind to DNA and they turn genes on or turn genes off. And so what we have here is a list of genes that have 2 properties. The first is that estrogen binding turns them on, and the second is that their proteins govern this very simple lordosis behavior. And we have a whole list of them. And the only one that I'll talk about, use as an example, is estrogen turning on the gene for the progesterone receptor. And that's especially interesting because the progesterone receptor is also a transcription factor. So you have a ligand-activated transcription factor turning on another transcription factor. And to speed things up a little bit, and we're going to ask how that happens. But first I'll show you that however it happens, it must be important. Because when you put an adeno-associated virus with a small interfering RNA, siRNA, against estrogen receptor alpha, you bring lordosis behavior from normal behavior down to 0. Here's another experiment, normal lordosis behavior down to 0. And this AAV, adeno-associated virus, is put just in the ventromedial nucleus of the hypothalamus. In those cells that I showed you, bind estrogens. And likewise in the preoptic area of maternal behavior, if you put in this adeno-associated viral vector bearing the small interfering RNA against ER-alpha, estrogen receptor alpha, the latency to doing maternal behavior, that is to going out and picking up the baby mice, goes to very, very high from very, very low. And the amount of maternal behavior goes from normal levels down to near 0, not quite 0. So this transcription factor, ER-alpha, is important for really all aspects of female reproductive behavior. And how does that happen? You probably know that there's no such thing as naked DNA. DNA is always covered by chromatin, which are proteins, and an important part of these proteins are called histones. And we're dealing here with histone H4. And what we've shown is that estrogen treatment acetylates, that's a pro-transcriptional step, acetylates histone H4. And the way it does it is in these cells on the ventromedial hypothalamus. Here's double immunocytochemistry for, here's the acetylated histone, I don't know if you can see that, here's the ER-alpha, and here's the merge. So every yellow cell here in the ventromedial nucleus of the hypothalamus, this is the bottom of the brain right here, every yellow cell is expressing both the estrogen receptor alpha, having responded to estrogen, and the pan-acetyl H4. And here's a whole bunch of molecular experiments in which, as a function of time after estrogen treatment, the pro-transcriptional marks, like this one right here and this one right here, go up. And the transcription repressive marks, I can't explain that. But here it goes down. 6 hours, about the time that PR, progesterone receptor, will be induced. The anti-transcriptional mark is right here. The one example that I want to give out of many is a so-called chromatin immunoprecipitation experiment. The basis for that is right here, that after estrogen treatment, both forms of the progesterone receptor, the so-called big form and the little form, go up significantly 6 hours after estrogen treatment, and here's how that happens. Just to show that you can bring an entire behavior down to the level of hormone-regulated transcription. I don't know how many of you are molecular biologists, but here you have the progesterone receptor promoter. Here are the transcriptional start sites. And what we did was we used a so-called chromatin immunoprecipitation technique, CHIP, in order to study the condition, the chemical condition of these histones on the promoter. And here are 3 sites on the promoter where we did it, which are mapped here, here, and here, 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 and here. And here are 2 transcriptional marks that we studied, pro-transcriptional marks, acetylated histone 3 and trimethylated histone 4. Trimethylated lysine 4 on histone 3, sorry. And here's the estrogen effect. No estrogen, estrogen, no estrogen, estrogen. And the bottom line is that yes, estrogen treatment does lead to histone modifications, which will then lead to transcription. But the nature of the change depends upon the exact histone mark you're studying and the promoter segment that you're studying. 3 different promoter segments. And this is the work of a molecular biologist in the lab named Katuna Gagnidze. And this does matter for behavior because when you promote transcriptional acetylation, like right here with this drug, TSA trichostatic acid, and you put this just in the ventromedial nucleus of the hypothalamus, lordosis behavior is increased and rejections by the female, which is usually the female kicking the male in the ribs, are significantly decreased. So what we've got is a specific gene, estrogen receptor alpha, and a specific group of neurons, ventromedial hypothalamus, with effects that read through a determined circuit, which I showed you a few slides ago, to regulate a specific behavior, this extremely simple behavior called lordosis. And the encoding of this chain of behaviors by estrogen receptor alpha shows how mechanisms for instinctive behaviors are harmonized with preparations for reproduction elsewhere in the body. And what we're doing now is this chromatin immunoprecipitation technique, CHIP, not only for PR promoter, but for other genes like oxytocin promoter, oxytocin receptor promoter. And I also have to tell you that these hormones not only work through the genomic mechanisms, which I've just given you a little flavor for, but also their membrane-initiated mechanisms, which are an entirely different can of worms.
So I have a naive question, actually.
Yes.
So since reproduction is seasonal,
Yes.
how is the expression of the estrogen receptor alpha controlled seasonally?
So the question is, since reproduction in many animals is seasonal, how is the transcription of ER alpha regulated seasonally?
Yes.
And the simple answer is I don't know.
Okay.
The somewhat more complicated answer is that some animals prepare themselves for the cold weather where you don't want to have your babies by generating a lot more thyroid hormone action in the brain. And there's a lot of thyroid hormone action in the brain. And it turns out that thyroid hormones fight against estrogen hormones as they affect genes. Like on the DNA, the estrogen response element, the place on the DNA where the estrogen receptor binds as a dimer and turns on the gene, is AGG-TCA, and then 3 bases. It doesn't make any difference what they are, but they have to be 3. And then the reverse symmetry, TGA-CCT. And that's the estrogen receptor is binding like that. It turns out that a thyroid hormone receptor can bind to an ERE half site, and therefore could conceivably, and we've proven this, can interfere with ER alpha action. So it could be that one of the mechanisms, not necessarily the only one, by which seasonality is regulated is not by ER alpha going up or down, but by ER alpha having an opponent or not having an opponent. And one of the names of the proponent could be thyroid hormone receptor alpha. But that's not really an adequate answer to your question. It's simply a molecularly interesting answer. And we're working on that right now. But the papers were by Vasudevan, the Indian name, Vasudevan, in 2001 was the first paper along those lines. But we're doing it right now. So let's wrap it up in the final 2 slides. A moment of introspection in your own lives will tell you that there's a non-trivial relationship between being generally alert and aroused and being sexually aroused. And it turns out that that's true in experimental animals, and we can see the mechanisms for it. And first we'll talk about the genetic female, XX, and we'll talk about the work. Usually when I'm talking in a different meeting, I'll have a whole bunch of slides from my former postdoc, whose name is Jessica Mong, and she's now a professor at the University of Maryland School of Medicine. Among humans, women, pharmacologically heightened, for instance, by methamphetamine, generalized arousal elevates this is female rat courtship and sexual behavior. It's true in women, but she can do it experimentally with methamphetamines and female rats. And so if you look up her papers, Jessica Mong, you'll see the evidence. Secondly, when we genetically lower, with that breeding project I told you about, generalized arousal, we reduce mating behavior. Thirdly, we know exactly how it works. Generalized arousal transmitters, like histamine and norepinephrine, depolarize the neurons which are essential for lordosis behavior. And prostaglandin D, which puts you to sleep, is reduced by estrogen. Prostaglandin D inhibits lordosis behavior. So we know 3 of the mechanisms. And finally, the actual female behaviors themselves require high generalized arousal, because what the female is doing is not just walking along with a normal locomotion. What it's doing is it's running very fast and then coming to a stop, and very fast and coming to a stop. And what that's doing is that even if the male doesn't know what he's doing, he's sniffing the female's behind, the vaginal odors. And when the male darts away from him, he follows her, and when she comes to a stop, he bumps into her in a perfect position for mating. So even a stupid male that doesn't know what he's doing crashes into the female by accident, and it's perfect setup for mating. And of course, that's what rats do. In New York City, it's estimated that there's 1 rat for every human being. In the male as well, just look at the left side of this slide. What we did is we bred, as you know, for heightened arousal or for lower arousal. And whether you tally your data according to the parents in the high arousal line versus the low arousal line, or the offspring in the high arousal line compared to the low arousal line, when you look at the number of mounts before the male gets the first penile insertion, it's called an intermission. The high arousal animals, the red bar here and the dark green bar here, are like overexcited teenage boys. Boys that are about 14 or 15. The testosterone has just hit the brain. They're going nuts every time they see a female. And they leap all over the female, but they don't know how to actually get a penile insertion. And so the high arousal animals are mating like crazy, but they're not being successful. Once they are successful, to the disappointment of everybody, they ejaculate immediately. And so this is the impact of arousal on male mating behavior in mice. The data are here, and this was all published a couple of years ago. So in females and in males, females from the Jessica Mong lab at Maryland and the males from our lab at Rockefeller both indicate that the forces supporting generalized arousal contribute towards sexual behavior. So summarizing the entire talk, we're trying to go for the simplest, most primitive, most elementary brain function that there is. If we were doing geophysics, we'd be trying to study the magma of the Earth. And so we hypothesize it, we define it, we assay it, and we know some of its anatomical, physiologic, and genomic mechanisms. We're now trying to highlight its quantitative properties, and we may be stumbling in doing so. And we're interested in its application to medicine and public health, depression, as Partha said, chronic fatigue, more frequent in females than in males, and so forth and so forth. With respect to heightened states of generalized arousal, as it applies to sex behavior, we've determined how sex behavior works, how it's regulated, and we're trying to study its relationship to generalized arousal mechanisms. We're sure, but based on those last couple slides I showed you, that somehow generalized arousal contributes to female and male sex behavior. In the female, we actually have some of the mechanisms already. And what we want to do is to hope for applications of generalized arousal to medical problems, especially those ones that I listed at the very beginning of the talk, and especially those with large sex differences. Females in the States, females have more depression than males in the ratio of 2.7 to 1. In Denmark, the ratio is 6 to 1, and I'm trying to figure that out. And so I was wandering around Copenhagen looking for depressed females, and I didn't see any. And it turned out it's July. And as far as I can tell, there's no Danish females in Copenhagen in July. It's all tourists from other countries. I was at a meeting there. And so I didn't see them. ASD. What is that? Can't remember.
Spectrum disorders?
Atypical sexual disorders.
It may be that. No, there's a different. Anyway, we'll forget that. ADHD.
Autism spectrum disorders.
It's autism spectrum disorders. Yeah, that's right. This is more frequent in males. If you're a low IQ male, there's no sex difference between males and females. But if you're a high IQ male, then the ratio is 9-to-1. Asperger's males to females. So probably about half the males in this audience have Asperger's. And, or at least we're surrounded by Asperger's, especially in our math classes. ADHD, attention deficit hyperactivity disorder, is more frequent in males than females. And so in terms of getting biological sweet spots for explaining these things, the ones with sex differences are probably some of the best. And finally, the guy that did all the electrophysiology is Eugene Martin. He likes the movie star Harrison Ford, so he has his Harrison Ford hat here. Lee Min-Kao has been with me ever since he got his PhD from Caltech. The virologist that produces the AAV vectors is right there. The circadian expert is Sergei Musatov, who comes to us from St. Petersburg. Ana Ribeiro is Portuguese. She did a lot of the circadian work. A lot of the sex behavior work was done by, again, Murakami. He's back in Japan, and so forth. So at any rate, I'm afraid I've outworn your patience, but we've had some good discussions. And if there's any other points, I'm happy to try to address them. But I have a feeling that I've outlasted you. Yes, please. Yeah.
So, for the sexual behaviors, so one challenge now is to sort of show or to argue that this is an example of this general arousal system.
Mm-hmm.
And this is really the core, the guts of it, right, how it's operating.
Yes.
But now, sexual behaviors often rely on highly specialized structures.
Yes.
I was first thinking about chemical sensing structures that are really sort of placed outside the generic processing structure.
Yes.
Right? So I mean, it's absolutely marvelous work, but there is a possibility that you have dissected a very specialized behavioral subsystem.
Not only a possibility, but a certainty.
Right,
Yeah.
that might therefore not really tell us a lot about these more generic behavioral functions under point one. So
Absolutely. So the question is, what about, and this could apply to any behavior there is, what about the impact of these more generalized forces, like GA, which I've generalized arousal, which I've been trying to talk about, and all of the specialized apparatus which goes into the proper biologic regulation of any animal behavior or any human behavior. And so remember, if we think of sexual behavior as an equation, it's likely to have a term that's called generalized arousal, which influences it, but there are all these other terms. What about the motor capacity of the animal? What about the sensory capacity of the animal? What about the presence of pheromones? What about the ability of the animal to process the pheromones? Because after all, in a mammalian brain, the pheromone probably starts out in the vomeronasal organ, which is also present in human beings, by the way, and then it goes not to the the regular olfactory bulb, but to the accessory olfactory bulb, and then to the medial nucleus of the amygdala, and then to the hypothalamus. And so at all of those parts, there's specialized neuronal mechanisms, as you said, that have to do with the specific causation of that particular determinant of that particular sexual behavior. And I not only admit all of those things that you just said, but I revel in them because it shows us the specificity of behavior, whereas most of this talk has tried to emphasize exactly the opposite. Yeah.
Other questions? I have one. Actually, a couple. One is the arousal, let's say the generalized arousal system being an accelerator. Is there an active brake in the brain? And second question is that we've heard about all the lovely homologies between invertebrates and vertebrates. Would either of you like to comment about invertebrate arousal? Yeah.
So in terms of the brake, B-R-A-K-E, I think we're talking not only about the GABA neurons in the medullary reticular formation, which are likely to be very important, and so far in our electrical recordings, they're just the little spikes, and then we're recording the big spikes, which are from the giant cells. And they're likely to act as one brake, but not the only brake. And as a matter of fact, a few slides ago, I showed a different brake, which is called prostaglandin D. If you transfer the cerebrospinal fluid from a sleeping goat to an awake goat, the awake goat will immediately go to sleep. This is the work of John Pappenheimer at Harvard Medical School. And it turned out that the active component of the sleeping goat's cerebrospinal fluid is prostaglandin D. And so they're elaborated by glial cells of great interest to drug companies, elaborated by glial cells in the forebrain, and also elaborated by a specific group of neurons, which Jessica Mong, that woman that I talked about, and I discovered in the preoptic area. Prostaglandin D would be an example of your point at the chemical level, bringing the animal to sleep. Prostaglandins are wonderful compounds. About the homologies, I really...
Follow on that question a little bit though.
Yes.
There is no actual neural system set up to bring it down, so to speak.
Yes.
It's just like the sympathetic, parasympathetic, in the
yes.
peripheral nervous system,
And
the central nervous system.
in terms of neural systems that are generated, and it is their business to bring us down. The speaker who you wanted to come here and could not come, named Clifford Saper,
Yeah.
S-A-P-E-R, at Harvard Medical School, talked about the push-pull regulation, and he has discovered mutually inhibitory neuronal systems, which either bring the animal's arousal up or bring it down. And so if you look at the work of Clifford Saper from maybe 2005, and a series of papers since then, you'll see an example of what Parth is talking about. And he was sorry that he couldn't come. Now, about the homologies, I really liked that slide that Nick showed a couple days ago, where, with the basal ganglia, for example, but 5 or 6 other principles. Until you discover all of the details of how they work, which we haven't quite done yet, you can't prove that the homologies are not a very elegant example of wishful thinking. Would you agree?
Yes, I think so.
Yeah.
I think one needs a very large data set to be able to suggest
to prove it.
Yeah, not to prove, but at least to suggest that it cannot possibly be derived by conversions.
Right.
Otherwise, convergence equals wishful thinking, I suppose. But what it really impresses me, what intrigues me is that, for example, with respect to the mouth cells, which you're not talking about, but you made the suggestion that these giant neurons are homologous to mouth cells. The mouth cell organization of a fish is, I would say, I use the word identical to that of a Drosophila.
Mm-hmm.
There is exactly the same organization, exactly the same conversion, exactly the same conditions of activating the system. It's quite remarkable. And then in the fly, we also discovered these very, very large, very few huge neurons that send dendritic and axonal aberrations throughout the brain, and also very, very large descending pathways to the thoracic ganglia. They've never been described before. We have no idea what they're doing. We're tapping on them, and they're full of peptides.
Where in the brain are they?
They are actually in the protocerebrum. And these are very, very peculiar cells, and I'm intrigued by this talk because it's sort of inspirational, and I have to go back and look at these in much more detail with regard to the activation by novel stimuli, maybe the novelty detectors of the kind that provides the arousal mechanism.
So before giving an inclusive answer to Partha's second question, I'll just mention that we could talk about other animals as well, like Aplysia. And Aplysia doesn't have the organized brain. The Aplysia has a series of ganglia which talk to each other. But if you want to study arousal of behavior in Aplysia, you might look at the work of Irving Kupferman, and talking about the stimulation of feeding behavior from the central ganglion of the Aplysia. But coming back to Partha's question, I think that neither Nick nor I believe in accidents or believe in coincidences. And therefore, the fact that he can, in an efficient way, in an elegant way, pair these systems up against each other, next to each other, between his systems and the mammalian systems, I don't think it's just an accident.
But I'm asking a specific question. I'm asking, are there these 5 systems that you showed, norepinephrine, dopamine, etc., are there known homologs?
Yes,
just curious to know
there are. Dick
if
Nessel,
they're organized.
Yes, Dick Nessel, University of Stockholm, has done beautiful work showing that there are essentially the same systems in the fly.
Using the same molecules,
Yes.
but are they spatially organized? In some
Yes, I think they are. Yes.
Because that would be a very natural way to look for homologies between
Right.
brains.
A pleasing way to talk about it is that once a Mother Nature has evolved a system that works, she doesn't or he doesn't necessarily throw it away. He might decorate it in many, many different ways with regulatory factors that are present in one animal but not another. But if it works, you don't discard it.
So to follow up on this question, with the Maunder cells, maybe that's not the right analogy, right? Because in the reticular formation, you'll find other types of neurons that actually do control discrete behaviors.
Mm-hmm.
So maybe they might be closer to this Maunder cell analogy, and now the new neurons that Nick just mentioned might be closer to these more arousal regulators that you're talking about.
Sure. In pointing out similarities or parallelisms between different systems, we're never trying to say that they're identical. A rat is not even a mouse, and one mouse strain is not another mouse strain.
The Maunder cell NGC match might be less clean than it looked initially.
Might be, yeah, I agree. Yeah, I wouldn't want to push it farther than the data would let us. Yeah.
Any other questions? If not, let's thank Don for this beautiful talk.